Good afternoon, dear participants. My name is Emine Erda from Nearest University Faculty of Pharmacy from Pharmaceutical Chemistry Department. Today I'm trying to introduce our research about in silico evaluation of ligands targeting the caspase-1 inflammasome pathway in the management of endodontic diseases. And also our molecular docking process, our results, and how we can manage these well-known inhibitors in the endodontic diseases in the future. First of all, this is our campus. I want to show you our campus first. This is the Faculty of Pharmacy. Here is the nearest university hospital, and this is the Faculty of Medicine. So for the introduction part, I will try to give an introduction about general information regarding to the endodontic diseases and what is our aim of doing that molecular docking study. As a general information, endodontic disease is defined as the immunoinflammatory response of the pulp or periapical tissues against endodontic pathogens, exhibiting various virulence factors associated with the host infection. As an example of these virulence factors, we have LPS and LTA, which are lipopolysaccharides, and the lipoteichoic acid. They are considered to be the most important virulence factors because they have the highest immunogenicity in terms of pulp or periapical tissue damage. The knot-like receptor family containing a pyrine domain 3, which is NLRP3, inflammasome pathway is crucial for the dental immune system, and it includes the activation of the dental immune response. That's why that inflammasome pathway is important for the endodontic diseases, because in the inflammation pathway, this inflammasome pathway plays a very important role in the dental immune system and the immune response. Here's a general cycle, the evaluation of ligands targeting the knot-like receptor 3 and caspase-1 inflammasome pathway. Here you can see the main evaluation cycle. When you look at that cycle, you can see that there is a signal 1 and signal 2. Signal 1 is a priming signal. One of the pathogen-associated molecular patterns, as an example, we have P. gingivalis, activates the transcriptional factor NFKB and thus increases the production of knot-like receptor 3. A procytokine, which is interleukin 1 beta, and also the another interleukin, which is interleukin 18. Another part here, there's a signal 2, which is an activation signal also. One of the pathogen-associated molecular patterns, such as ATP, activates the knot-like receptor 3 inflammasome via the P2X7 receptor. Here you can see that hypoxia helps to activate the NLRP3 inflammasome. After the activation of that inflammasome, activated caspase-1 produces the active form of interleukin 1 beta and interleukin 18. The activated caspase-1 also cleaves gastermin D at the N terminus, thus forming the inducing the formation of pores and inducing the pyroptosis of the cells. So that's the general evaluation cycle of ligands targeting that inflammasome pathway. What is our aim when you look at that pathway? And we have already said that the inflammasome pathway is important in the dental diseases, the endodontic diseases. So, what is our aim of doing that study? Our aim is first, evaluation of the binding energies and interaction of different inhibitors known to play an important role in inflammation through the direct interaction of not like receptor 3 binding sites or through the inhibition of caspase 1 active site. So, our second aim is the selection of the most effective combination for use in endodontic treatment or as a complementary combination for the main strategy of endodontic treatment. So, what are our methods while performing this study? In this study, human not-like receptor 3 model was constructed by the loop modeling methods using computer-aided programs. And then after that loop modeling process, we did a molecular docking which is also performed using computer-aided programs. We used actually modeler program for the loop modeling, and we used the Autodoc Vina for the molecular docking. While using these programs, we measure the binding affinities of these well-known inhibitors and the inhibition constants, and we generate the ligand protein interactions of the selected ligands, and they are calculated and investigated by molecular docking simulation against the inflammasome not like receptor 3 and caspase 1. When we look at our results here, there is a 3D representation and also 2D representation. We already used different ligands, which are already known to be effective, not like receptor 3 inhibitors, such as CY09, MCC950, tranilast, oridonin, MNS, and OLT1177. These are well-known inhibitors which are discovered, and we perform this study in order to get their binding affinity and compare their binding affinities and the inhibition constants to get the most effective one in terms of desirable binding affinity and the inhibition constant. And in the table, we have also the amino acid residues for each of them after molecular docking process. So as a result, we got the most desirable binding affinity and inhibition constants when we compare these inhibitors. CY09 is found to be the most effective inhibitor for not like receptor 3 model. So for the not like receptor 3 model, we have the ligand CY09, which is the most effective one. And then when we continue with our results, we also test the caspase 1, human caspase 1 activity. And we have also different inhibitors which are called VX765 and partenolide. These are well-known caspase 1 inhibitors in the literature. So we took them and made the molecular modeling by downloading the X-ray crystallographic structure from protein data bank And then we perform the molecular docking process using these ligands. And then we compare the ligands interaction with the binding pocket of caspase 1, and we got the 2D and 3D binding interactions, as you can see here on your right side. The green dotted lines denote the hydrogen bonds between ligands and amino acids, whereas the pink lines here denote the hydrophobic interactions. And as a result, between these two caspase 1 inhibitors, we found that VX765 is the most desirable binding affinity and inhibition constant when we compare it to partenolide. And now, as a caspase 1 inhibitor, we select VX765. As a conclusion and our future aim, as a summary up to here, the ligands CY09, which we found in the study that it is the most desirable, has the most desirable binding affinity and the inhibition constant. So we select that ligand by comparing with the other ones. And VX765, which is the caspase 1 inhibitor, when we combine them, we thought that they have the most desirable values so that they can be used together in combination to prevent inflammation in the treatment of endodontic diseases. And we hope that this study may shed light on the design and synthesis of novel knot-like receptor pyrene domain containing 3 inflammasome and caspase 1 inhibitors for use in LPS or LTA induced endodontic diseases. Thank you very much for listening. If you have questions, I can take. By the way, this is our team. I want to show you our team, which has many efforts. Everybody has many efforts in this study. I would like to thank each of my professors, associate professors, and assistant professors. Thank you very much.